The overall goal of targeted next-generation sequencing is to elucidate the genetic determinants of various constitutional diseases by focusing on genomic regions of particular interest.
So this method can help us answer key questions regarding a disease's genetic etiology, particularly when there are previously known genetic associations. This technique is highly efficient. It produces millions of reads in a short period of time. We get them at a relatively low cost. There's relatively low computational burden, especially when we compare it with other next-generation sequencing approaches.
The implications of this technique extend toward the diagnosis of neurodegenerative diseases, which are phenotypically and genetically heterogeneous, but have many known associated genetic loci.
Though this method is particularly directed towards neurodegenerative diseases, it can also be applied to various other constitutional diseases with previously identified genomic regions of interest.
Generally, individuals new to this method will struggle because the bioinformatics processing required for final rare variant analysis can be computationally intensive and create many sources of error.
In this procedure, human blood samples are collected in 3 4 milliliter EDTA K2 tubes to provide a total volume of about 12 milliliters. Centrifuge the blood samples at 750 times gravity for 20 minutes. This will fraction each sample into an upper phase of plasma, a thin middle phase of leukocytes, and a bottom phase of erythrocytes. Pipette the plasma off of the top of the blood sample using a disposable transfer pipette. Dispense into multiple 500 microliter aliquots